Lasers have been used successfully in science and technology for many years. We are going to tell you how to measure the dimensions of microscopic biological objects using a laser.
The thickness of these objects can be compared to the thickness of a human hair. What's more, the accuracy of these measurements is quite high. We carried out all the scientific research in the physics laboratory in Moscow private school New Education. What objects can be measured with a laser? My classmates have researched the size of erythrocytes, red blood cells of humans and frogs, the diameters of eyes of dragonflies, bees and butterflies, the structure of microscopic sections of plants and onion peel, the thickness of veins of dragonflies' wings.
My work is dedicated to laser diffractometry of microstructure of polyporous fungi hymenophore. Polyporous fungi can be found on all continents. The majority of polyporous fungi harm trees on which they grow. Some of them are used to prepare drugs. Some species of polyporous fungi are edible.
In these slides you can see the polyporous fungi which we have collected in Moscow forest parks. You can see the great variability of mycothallus of different species of polyporous fungi. Mycothallus within each species can vary too. One of the identifying features of polyporous fungi species is the structure of its poriferous layer. This layer is called hymenophore. We can examine hymenophore structure of polyporous fungi in the next slide. In these photos we can distinguish cylindrical pores. These pores can be several centimeters long. We can see orifices of the pores on the lower surface quite well. At the bottom of the slide you can see the photos of microscopic sections of hymenophore. Using a microscope, we can see that the orifices of the pores of Fomes fomentarius are practically circular. The diameter of pores is an important identifying feature. However, only the average number of pores per 1 mm of hymenophore surface is indicated in reference books. This value is usually calculated by routine counting using a magnifying glass or enlarged photographs. Now we can formulate our hypothesis. The diameters of hymenophore pores of polyporous fungi and their linear density can be determined by means of the diffraction method with high accuracy. So, the purpose of this project is to find out if the laser diffractometry method can be effectively used to identify species of the polyporous fungi. We had to solve some problems which I'll tell you about later.
What's the essence of the method? The experimental setup is simple. A microscope section of hymenophore of polyporous fungi was placed in the laser beam.
As a result of the diffraction on the pinholes of hymenophore, it's possible to observe a diffraction pattern on the screen in the form of alternating light and dark patches. For different diffraction patterns, we worked out the formula, with the help of which we calculated the distance between pores and the diameters of hymenophore pores. One of the most important problems was to prepare a sample. The diffractometry technique can be used for examining only those polyporous fungi, the pores of which are almost cylindrical. That was the group of 6 species we have examined. The most difficult species to examine was Trihapton pergamenum. Its pores only vaguely resemble a circle. What did we observe during the experiment? When the laser beam got onto different parts of the surface, we observed different diffraction patterns. We managed to identify two different kinds of diffraction. In this slide, you can see the diffraction pattern in the form of alternating black and red rings. Such diffraction occurs in the parts of section where pores are set irregularly. With the help of these patterns, we managed to calculate the diameters of pores. We measured the distance between the sample and the screen and the diameters of dark rings. In these photos, we can see the diffraction patterns in the form of separate maxima. In these parts of the microscopic section, pores are set regularly. With the help of these patterns, we calculated the distance between the centers of the pores and the number of pores per 1 millimeter of the surface. We measured the distance between the sample and the screen, it's more than 4 meters, and the distance between the symmetrical maxima. The final experimental results for all studied species of polyporous fungi are represented in this table. In the second column, we can see that the diameters of pores of different species are quite different. It's the first time these results were obtained with such a high accuracy. Our result of the measurements of linear density of pores coincide with our calculation on photos and on data from reference books. The detailed results of measurements of the pore diameter of one polyporous fungi species are represented in this table. You can see that the accuracy is very high. The error is less than 7%.
In conclusion, I would like to say that in our project we prove that the usage of the laser diffractometry technique is of great practical interest. This technique has a lot of advantages, as well as some disadvantages.